Hello everybody, you are welcome. Today we will solve paper 4 variant 2 October-November 24 Cambridge IGCSE Biology. Okay, let's start. In this exam we will focus on 6 questions. The first one deal with classification and diet. Second one, gas exchange. In question number 3, we focus on excretion and diabetes type 1. In question number 4, sexual reproduction in plants. In question number 5, codominance. Last one, we concentrate on nitrogen cycle. Let's start. The first question. Letter A, organisms from the genus Chlorella are protocysts. State the additional information required to name Chlorella according to the binomial system. Look here. This is tiger. This is the common name or English name. Okay. When we deal with binomial system, we need two names. The first name refers to the genus and start with capital letter P. The second name refers to the species and start with small letter T. Okay, the same also when we deal with Chlorella as genus. So where is the species? So the additional information required to name Chlorella according to binomial system is the species name. It's okay. Now for letter B, figure 1.1 shows the structure of an organism from the genus Chlorella. The question here, single I, identify two cell structures in figure 1.1 that indicate that Chlorella is not prokaryote. That is not a prokaryote. Prokaryote like bacteria. Bacteria lacks nucleus. Its genetic material DNA spread in cytoplasm and lack also membranous organelles, no chloroplast, no mitochondria, no rough endoplasmic reticulum. Okay, so here I need to mention two cell structures indicate Chlorella is eukaryotic. It contains nucleus. Okay, it has chloroplast. Okay, cup shape chloroplast. Letter double I, identify two cell structure in figure 1.1 that are found in both fungi and protocysts. Chlorella here, and this figure for yeast cell as an example of unicellular fungus. Okay, yeast cells or yeast cell. What about structures found in both fungi and protocysts? Yes, both contain cell membrane, surround cytoplasm, and both these two examples surrounded from outside by cell wall. So cell wall, cell membrane, cytoplasm, and nucleus. Any two from these four items considered correct answers. Okay. Now for letter triple I, state the names of two other kingdoms apart from fungus: prokaryote, protocyst. Here three kingdoms. Okay, what the remaining two kingdoms? Yes, plant kingdom and the animal kingdom. It's okay. Now for letter C, Chlorella is sold as nutritional supplement. Spirulina is another nutritional supplement. Okay, table 1.1 shows some nutritional information for Chlorella and spirulina supplements and the recommended daily intake for some nutrients. What about some nutrients here? Vitamin C, calcium, iron. Okay, with second column, mass of nutrients, how many milligrams per 100 gram of supplement for both chlorella and spirulina. Here the average recommended daily intake in adults, milligram per day. Look, what about the question here? He asked about one tablet contains 5 gram of chlorella supplement. One tablet like this. Okay, its mass equal 5 gram. Using the information in table 1.1, calculate the number of tablets of chlorella supplement a person needs to take to provide the recommended daily intake of iron. So the question here about iron. What about iron? Okay, here iron for 37 milligram per 100 gram of supplement. Each 100 gram of chlorella provides 37 milligram iron, but each tablet in the question here, okay, with 5 gram as mass, provide how many milligram of iron? Provide x milligram of iron. So what about x? By cross multiplication, x equal 5 gram times 37 milligram, okay, divide 100 gram. Cancel gram with gram. Okay, the remaining result equal 1.85 milligram iron. This for each tablet, for one tablet. Okay, what about the daily? How many? He asked about how many, or calculate the number of tablets. How many tablets needed to cover the recommended daily intake of iron in adults? How many? Here, look, we need maximum recommended daily intake 12 milligram, but each tablet only provide 1.8.5, less than 2. So how many tablets needed to provide this recommended daily intake? Equal number of tablets equal 12 milligram divide 1.85 milligram cancel with milligram. Okay, so equal 6.486. But look please, 1 decimal place, give your answer to 1 decimal place. So we have to approximate this figure into 6.5 tablets daily. Okay, this supply whole recommended daily intake of iron, I mean 12 milligram per day. Okay, letter I. Explain the advantages of taking chlorella as dietary supplement rather than spirulina. Use the data in table 1.1 to justify your answer. Here's the table. Okay, what's the advantage of taking chlorella? Chlorella contains more vitamin C than spirulina. Also contains more calcium, more than found in spirulina, but little iron. Okay, little than in spirulina. Okay, so what's the advantage of taking chlorella as dietary supplement rather than spirulina? So we have to mention and to discuss these two items, only these two items, vitamin C and calcium. Okay, for vitamin C, chlorella contains vitamin C 0.74 milligram per 100 gram of supplement. Okay, vitamin C is needed for wound healing, improve immune system function, or prevent scurvy, bleeding gums, like in this figure. Okay, one of this of the symptoms of scurvy, bleeding gums, and also pain, joint pain. What about calcium? Calcium here more also. Calcium is more. Chlorella provide or contains more calcium, means 120 milligram per 100 gram of supplement versus 5.1 milligram per 100 gram of spirulina supplement. So What the importance of calcium now? Calcium is needed to prevent rickets, okay, and strengthen bones and teeth, okay. Now for triple I, state the name of one food that is principal dietary source of vitamin C. Fresh vegetables and fruits contains a lot of vitamin C. Look at this figure: citrus fruit, orange fruit, okay, green pepper. All of these items contains a lot of vitamin C. Okay. Letter D, chlorella is good source of protein. State the importance of proteins in active transport. Here, this figure shows the active transport of certain molecules. Here, active transport means movement of particles from area of low concentration to area of high concentration, okay, against concentration gradient by using energy. So here, what's the importance of protein? Where is the protein? This one. This called carrier protein. Why it is called so? Because it carry by its binding site this particle and rotate and move within the membrane by help of energy supplied by ATP from respiration, okay, to move these particles against its concentration gradient. So look please to this figure. Particle recognized by carrier protein by its binding site, and by the help of energy the carrier protein move along the membrane, okay, and release these particles towards the area of high concentration, this against concentration gradient, okay, and move again to carry another molecule and so on. So proteins act as carriers in membranes. They allow movement of molecules, ions, particles from low to high concentration using energy, okay, like this door, this rotating door use energy, using electricity to rotate, to allow movement of particles within the membrane. It's okay. Letter double I, state the name of the smaller molecules that proteins are made from. All proteins are made of basic units, basic bricks, building units called amino acids. Its structure like this. It contains carboxylic acid group, contains amino group from the other side, okay. So this is called amino acid structure. By binding this amino acids together by certain type of bond called peptide bond, it forms polypeptide chain or protein molecule, okay. Second question, to letter A, figure 2.1 is a diagram of the gas exchange system in humans. Look at this figure, trachea, X, Y, and Z. So the question here about, I think, label, states the names of parts labeled X, Y, and Z in figure 2.1. X, external intercostal muscles. Here, rib. This reps, between reps, two sets of muscles, external and internal. Here, external intercostal muscle, okay, for X. For Y, yes, this trachea, this bronchus, this tiny or bronchiole, Y, bronchiole. What about the sheet of muscle, okay, flat sheet of muscle called diaphragm, okay. Letter double I, the wall of trachea contains rings of tissue. State the name of this tissue and describe its function. Here the figure of trachea, and behind and parallel to it esophagus from behind. Okay, esophagus is muscular tube. It belongs to digestive system, while trachea belongs to breathing system. So name of this tissue, rings of tissue. This ring of tissue called C-shaped cartilage rings, or incomplete rings of cartilage. What its importance? What its function? Its function, support and prevent collapse of trachea. Supports or prevents collapse of trachea. The tissue called cartilage. Okay, now for letter I. State the names of two types of cells responsible for protecting the breathing system from particles. Particles like dust, like smoke, like pathogens, okay, like toxins, like spores of fungus. Okay, which two types of cells responsible for protecting us from these particles? Here, look. Yes, ciliated epithelial cells. This tissue called ciliated because it possesses cilia at its free end towards the lumen, okay, of air pathway, and the goblet cells in between that secrete mucus. So two types of cells I mean here: ciliated cells that sweep or move mucus along the lumen, okay, towards the throat to be swallowed or to be ejected outside through mouth, for example, okay. While goblet cells produce certain sticky substance called mucus that stick and adhere to pathogens and particles. Okay, by help of action of cilia, it moves away, carrying these particles outside of our breathing system. Okay. Scientists estimated the pressure and volume in the thorax during one breath. Figure 2.2 shows graph of the results. Here, letter A, letter B. Pressure in thorax, volume of air in thorax. So pressure and volume. Okay. Now, through time. This during one breath. The question here describe. He need description and explanation. The changes in the thorax that occur during section E only in figure 2.2. Through this stage only. What about pressure? Pressure, look, decreases, then increase. Decreases, then increases. This for pressure inside thorax. What about the volume of air inside the thorax at the same stage A? Increases, only increases. Okay, only increase. So this in description. What about explanation? Look please, pressure inside thorax decreases and then increases. Yes. Volume of air inside thorax increases. Inspiration occurs due to these changes in pressure and volume. Inspiration occurs. How come? What's the explanation? The diaphragm contracts and flattens. Contracts and move down. Look as the figure here. The diaphragm contract, so move down. What happens? The thorax increase in its volume. The thoracic cavity increases in volume. What about external intercostal muscle? Also contract, like diaphragm exactly. Contract. Okay, both help to increase the volume of chest cavity. What about external intercostal muscles? It allow reps to move or chest cavity or thoracic cage, move upwards and outwards. This increases the volume of our chest. Okay, external intercostal muscle contract while internal intercostal muscle relax. they are opposite in action, so they are considered antagonistic muscles. Antagonistic because they oppose their actions during the breathing mechanism. Reps move up and outwards, okay, so this increases the volume. Here, what about intercostal muscle during inspiration? External intercostal muscle contract. Rib cage, little, okay. Rib cage move upwards and outwards. What about diaphragm contract? Like what? Like external intercostal muscle. Diaphragm moves down. Thorax volume increases. Air pressure decreases than the air outside the atmospheric air. So air rushes into the lungs. This during inspiration. The reverse exactly when he asks us about expiration. The opposite is correct for expiration. Okay. Now for letter C. Complete the sentences to describe the effect of carbon dioxide concentration on breathing. During physical activity, during vigorous exercise, the carbon dioxide concentration of blood increases. This is detected by the brain. The brain detects the change in CO2 or drop in pH. The brain. This results in an increased rate and greater depth of breathing. What about depth? Depth, I mean this distance, the depth of breathing. Here, less frequent breathing rate, less breathing rate. Here more breathing rate, higher breathing rate. Here little depth, here more depth. Okay. The brain center in medulla oblongata or brainstem stimulated, or control center stimulated by CO2 increase or pH decrease in blood. Okay, so the brain send messages through nerves to both muscles, diaphragm and external and also internal costal muscles. Okay, diaphragm to contract more and more, also for external intercostal muscles, while send impulses to heart to increase heart beating rate. It sends this impulses to the pacemaker of the heart to increase its rate. Okay. Now for question number 3. Figure A, figure 3.1. Okay. Question 3. Question number 3. Figure A. Okay. Question 3. Figure 3.1 shows a kidney nephron and its associated blood vessels. Here, Bowman capsule with glomerulus inside, and the letter A, where blood entering inside glomerulus. Okay, and so on. Now, the composition of fluids A, B, and C were analyzed. 3 fluids here. Fluid A, fluid B, okay, after passing through glomerulus, and then fluid C is the last one. The table shows the results for 5 components of the fluids. Okay, component water, glucose, protein, urea, ions for percentage concentration in 3 fluids A, B, and C. The question here about state the names of fluid A and fluid C in figure 3.1. Fluid A, yes, blood or plasma or blood plasma. Okay, what about C? Urine. It's okay, right? For double I, using the information in figure 3.1 and the table 3.1, describe and explain the differences in the composition of fluids A, B, and C. When we ask about description, please believe in your eyes, your observations, and then make sense explanation of your observations. Okay, so here, what about the differences in the 5 components in the 3 fluids A, B, and C? Look please at the table. I think the major one, protein, found here in A, but nothing in B or C. Glucose found in A with the same concentration like B, while in C nothing, no glucose in C. Okay, let's start firstly. What about the functions of nephrons? Ultrafiltration takes place in glomerulus. Filtration takes place, filtration of blood occurs in the glomerulus. No protein in B. No protein in B or C. Protein only in A. Why? Because proteins are too large to enter or to be filtered through the nephron glomerulus or the glomerulus. Okay. No glucose found in C. No glucose found in C. Why? Because all glucose reabsorbed in the first coiled tubule here in this part. After filtration, it is found with the same concentration in B like A, 0.1, 0.1, while in this part, completely reabsorbed. All glucose is reabsorbed in the nephron. Okay, higher percentage of water urea ions in C compared to A and B. Urine contain high percentage of water, okay, than B and A. Okay, also contains higher concentration of urea 2 rather than 0.03 in B or A. Okay, also for the ions, this excess ions like potassium ions excreted in urine. So some water ions are reabsorbed in B while excess is passed to C. Urea is excreted into C. Here, to summarize this lesson completely about the function of nephron, here Bowman capsule where contains glomerulus inside. This is glomerulus, ball of capillaries. Blood is filtered under high pressure, ultrafiltration for waste excretion, urea, uric acid. Some useful molecules like glucose, amino acids, ions like sodium, like potassium. Okay, most water are filtered into the kidney tubules, passes as filtrate. Okay, what about this part? Okay, this part called first coiled tubule or proximal convoluted tubule. Useful molecules like glucose, amino acid, and vitamins. Most of water are selectively reabsorbed. What is meant by selectively reabsorbed? Means return back to blood again, back into the blood capillaries. Okay, the fluid passes or filtrate passes down, then up here. Okay, where what would happen here? Collecting duct collects the urine, collects the filtrate from many nephrons, and passes this fluids down as urine. Urine contains water as solvent, urea as waste. Most of urea excreted. Most of nitrogenous waste like uric acid excreted also. Excess salts like potassium ions and sodium ions if in excess. Okay, hormones, metabolites, drug metabolites, medicines metabolites, all excreted in urine. Okay, excreted in urine. What about here in this part? In this part of glomerulus, most of blood plasma filtered, small molecules filtered, while blood cells like red, white, and the platelets plus plasma proteins, albumin, globulin, fibrinogen, are too big, are too large to be filtered, so pass directly to the blood flow, okay, into venous blood. Okay. Letter P, outline how blood glucose concentration is controlled. Controlled means homeostasis done by negative feedback mechanism. Look, please. Outline, he asked about main concept about glucose concentration, how it is controlled in our body. At first, high blood glucose sugar after having a meal promotes, enhances or stimulates insulin release from pancreas. Pancreas senses the increase in glucose level after having a meal, so it secretes insulin hormone. Insulin hormone, okay, needed to allow glucose enter cells, enhance or stimulate glucose uptake from blood by all cells, especially liver and the muscle, for storage excess glucose in the form of glycogen. This happens in both liver and muscles. Okay, beside all cells stimulated by insulin to allow uptake of glucose to be burned or oxidized to release energy needed by the cells. Okay, so what happens to the glucose level after storage and usage and uptake? Lowers blood glucose level happen. Okay, return to normal, to set point or normal range. When we have fasting, okay, when we are fasting, low blood glucose or exercising, low blood glucose level in blood enhances or promotes or stimulates glucagon release from pancreas also. Glucagon as hormone secreted from pancreas during exercising or fasting. Glucagon stimulate the liver to convert glycogen into glucose. Why? To increase glucose level in the blood, so it returns into normal level again. This is called the negative feedback mechanism or homeostasis. Look, homeostasis, negative feedback mechanism occurs. So this control happens by homeostasis or negative feedback mechanism. Pancreas detects changes in blood glucose concentration. Pancreas secretes insulin and glucagon. Okay, insulin decreases blood glucose concentration after having a meal. Okay, insulin stimulates conversion of glucose into glycogen inside liver and the muscles. Insulin stimulates all cells to uptake glucose. Look at the action of insulin. How does insulin works? Here, insulin as hormone has receptors in all body cells. These receptors combine or complementary effect with insulin. Here, glucose in blood after having a meal, insulin is secreted. Insulin bind with its receptor, allow opening of channel needed for glucose entry. Okay, so glucose enter easily or uptaked by the cell when insulin is present. So insulin stimulates uptake of glucose and also stimulate conversion of glucose to glycogen in excess. Okay, glycogen is stored in the liver and the muscle. Okay, insulin promotes cellular respiration, stimulates the cell to respire to release energy. What about glucagon? Increases blood glucose concentration during fasting or during having an exercise. Okay, look, glucagon stimulates conversion of glycogen to glucose, the reverse. Okay, letter C, a gene mutation may be involved in the development of type 1 diabetes. Describe what is meant by gene mutation. Gene mutation means what? Means sudden or random change in base sequence of what? Of DNA. Okay, of DNA, like this. Here, normal gene, normal DNA sequence, okay, normal sequence of nucleotides or base pairing. What happens? If this gene expressed normally, it forms normal protein, normal enzyme, normal antibody, okay, anything in protein in nature, normal. What happens when the gene mutated? Change in DNA sequence, one letter or more. This called mutation happen. Okay, this form what? Form abnormal protein, abnormal enzyme, okay, or no enzyme or no protein. So this disease, genetic disease, due to gene mutation. Okay, now outline the treatment of type 1 diabetes. Type 1 diabetes, how the patients of type 1 diabetes treated. Okay, through what? By insulin pump or injections, like here, injections, pump, insulin pump. Okay, monitor blood glucose concentration every day by this sensor, okay, or by this strap or sensor also, in blood or in urine. Control carbohydrate intake, their diet, okay, increase more fibers, more protein, less carbohydrate, okay. Regular exercising. All four items, all of these items needed by the patient to control blood glucose level daily. Insulin injections, healthy eating and meal planning, fruits, certain type, not all types of fruits allowed, increase physical activity, regular exercising, blood sugar checks or regulation. Okay, now for question number 4, figure letter E, figure 4.1 shows diagram of two flowers from different plants of the same species or species. Describe what is meant by the term species. Species means what? Means a group of organisms that can reproduce, like the giraffes. Okay, a group of organisms that can reproduce, yes, to produce fertile offspring, like the zebras, group of organisms that can reproduce to produce fertile offspring. Now, like humans, all of these humans are fertile and they can marry together to produce fertile offspring. Okay, this is the term species. Now complete the diagram in figure 4.1 to show self-pollination and cross-pollination by drawing one arrow to show the pathway of pollen during self-pollination and labeling this arrow self-pollination. Drawing one arrow to show the pathway of pollen during cross-pollination and labeling this arrow cross-pollination. Labeling the names of the structures involved in pollination. Look at the structures. Okay, two flowers of the same species. Here for label X and Y, okay, and also this pollets, first pollet, second pollet, third pollet, here, okay. Now the anther, okay, the male reproductive organ where meiosis takes place to produce pollen grains. Here stigma to receive pollen grains during pollination. Here from anther to stigma of the same flower means self-pollination. self-pollination. He asked about drawing one arrow to show pathway of pollen during self-pollination and labeling this arrow self-pollination. Okay, this created now. Drawing one arrow to show the pathway of pollen during cross-pollination. For cross-pollination, pollen grains from any anther from the first flower passes and transfer to the stigma of another flower, okay, on another plant. This called cross-pollination. For triple I, states the function of the parts labeled X and Y in figure 4.1. Many functions for X. It is the ovary. It contains the ovules. It contains female gametes with one N chromosomes. It is the site of fertilization where pollen grain nucleus fuse with the ovule nucleus. Fertilization happens inside this part, okay, inside the ovary. Also, the ovary forms fruits after fertilization. Certain changes happen where the ovary swell, okay, become larger and bigger to produce fruits, okay, forms fruits and seeds inside. And the seeds inside, as we know, ovules change into seeds and ovary changes into fruits, okay. What about Y? Y is green, often green part, protects the flower when it was a bud. Look, before blooming of the flower, something green in color cover or surround the flower. This part protect the flower when it was a bud, okay. Look at this video. Okay, where seeds germinate to form seedling, then plant become mature, okay, seedling, young plant, then large tree. It produce flowers pollinated by any type or any means, like insect pollination, for example, okay, where pollen grain transferred. Then what happens to the ovary inside? Swollen and form fruit. Inside the fruit, many seeds, one or more seeds, okay. Now for letter B, explain why self-pollination that results in production of offspring is a form of sexual reproduction and not asexual reproduction. Self-pollination that results in production of offspring is a form of sexual reproduction and not asexual reproduction. Although one parent is needed or one parent only responsible for self-pollination within the same plant, for example, same tree, same plant, okay, it is still considered sexual reproduction, not asexual. Why? Because of sure this involves gametes formation, pollen and ovules are formed, okay, each one with one N chromosomes, haploid cells, okay, involves haploid cells, one N chromosomes. Meiosis takes place, this during sexual reproduction. Also fertilization takes place, okay. Offspring show genetic variation. Even fertilization takes place within the same individual, the same plant. Offspring show genetic variation. Why? Because during meiosis mutation happens, also crossing over, independent assortment of chromosomes. All of this cause pollens and ovules are different, okay. This produce genetic variation within offspring. Okay. State the type of environmental conditions that hydrophytes are adapted to live in. Hydro means water. Hydrophytes means plant that live in water. I mean aquatic plant. Aquatic plant like this. Water lily, for example. Okay. So, state the type of environmental conditions that hydrophytes are adapted to live in. Water or aquatic environment. Where certain adaptation in structure, like a lot of air space is found in stem and root to allow gas exchange easily within the water. Okay. Now for number 5. Figure 5.1 is pedigree diagram showing the inheritance of blood group in one family. B and A. B the husband, A the wife. Okay. Where their offspring 3, 4 and 5, and their son number 5 married the female number 6 to produce offspring in second generation 7, 8 and 9. This family pedigree. See the number of people in figure 5.1 with XY chromosomes. Here the male with XY chromosomes, female with XX. Okay, he asked about XY chromosomes in this figure. Number 1, okay, son number 4, son number 5, this individual number 7. Okay, how many individuals now with squares with male features? 4. So the number of people equal 4. Okay, number of people with XY chromosomes equal 4. What about only one IA allele? IA allele. Okay, IA allele, look please. Here for the mother A to produce AB in this individual, so both parents must be heterozygous. So this genotype here, this female with genotype IA IO, here only IA, second allele IO. Okay, he asked about only one IA allele, this female number 1. Here also IA IB, only one IA allele. What about this one? Okay, offshore, as all offspring are O group, it means what? It means this individual IA IO, IA IO. Why? Because this heterozygous individual, okay, produces all offspring, all has offspring O group. Here, as you know, this recessive IO. Okay, to produce IO here, the husband or the father must be IA-IO. So how many individuals with IA allele? Number 2, yes. Number 4, yes. Number 5, yes. So number of people with IA allele equal 3. Okay, equal 3. Now for letter B, explain how figure 5.1 shows that blood group is an example of discontinuous variation. Discontinuous variation. Limited number of phenotypes. All populations throughout the world of humans belong to either A, B, AB, or O. Okay? No intermediates, no average, no mean. It's qualitative results. Okay, like here, blood group either A, B, A, B, O. No intermediate in between, no average, no mean. Okay, that's qualitative result. So it is discontinuous variation. Okay. State one example of discontinuous variation plants. Discontinuous variation plants like what? Like seed shape, like seed color. Here seed shape, round, smooth, round seeds of peas. Here wrinkled or green and yellow. Green and yellow, no in between, either green or yellow. Okay, here wrinkled or smooth, all of the smooth. Okay, round, here wrinkled, no intermediate between these two features or these two characteristics. Okay, what about letter D? Explain why the inheritance of blood group is an example of codominance. Codominance. What is meant by codominance? Means the heterozygous individual possess two alleles with the same power and responsible for appearance of certain character. Okay, the character or the trait of heterozygous individual is created by two different alleles with the same power. Okay, so blood group is an example of codominance. IA and IB and IB alleles are both expressed in which individual? In blood group AB group individuals. AB blood group shows both alleles contributing to phenotypes, like here. AB, both alleles IA, IB responsible for appearance of this phenotype. The individual carry both antigens, antigen A, like this one, and antigen B on the cell membrane of red blood cells. Both are expressed to produce certain proteins found on the cell membrane. So that's good example of codominance in human. Blood group AB. Both alleles in heterozygous individual responsible for appearance of certain trait. Okay? Okay. Now for what? For this problem. Letter E. A person with the genotype IAIO has child with a person with the genotype IPIO. Complete the genetic diagram to determine the probability of the offspring having the blood group AB. Parent 1 and the parent 2. Blood group A and B. Okay. Both has a child with the genotype IPIO. Okay. What about this one? Here parental phenotype blood group A. Parental phenotype B. Here genotype IA, IO, IP, IO. Heterozygous A, heterozygous O. What about, heterozygous A, heterozygous B. Here how many gametes are formed? IA, look, IO. Two types of sperms. Here IB, IO. Two types of eggs. This probabilities of this parent to produce two types of gametes. The second one also two types of gametes. 2 times 2 equal 4. So the probability to found 4 individuals. Look of to offspring genotypes and offspring phenotypes. Here IA, IP. IA, IP. The first one. IA, IO. IO, IB, IB, IO. Okay, we start with alphabetical manner. IP, IO, and then IO, IO. What about offspring phenotypes? Offshore IAIB, AB. IAIO A, IBIO B, and IOIO O. Here, in 4 individuals in offspring, probability of the offspring having the blood group AB 1 in 4, I mean 25%, or 0.25 or quarter. Okay, 1 over 4, 1 out of 4 carry AB heterozygous blood group. Okay, AB. Now for number 6, last question about the flow chart in figure 6.1 shows one pathway of nitrogen as it travels through the nitrogen cycle. Nitrogen in the air, okay, nitrogen fixation by nitrogen fixing bacteria in the soil and root nodules. Nitrogen fixation happens in bacteria free in soil called the nitrogen fixing bacteria, and another bacteria found on the roots of legumes like beans. Okay, here nodules contain a lot of nodules containing bacteria that fix nitrogen in soil into nitrogenous compounds. Then uptake of nitrate ions and synthesis of protein by plants. The plant obtain nitrate ions by active transport or by diffusion, simple diffusion. What happens by eating? Okay, by absorption. Then the plants eaten by herbivore like cow, like rabbits. Deamination in animals occurs to form what? To form urea. Nitrogenous compounds in human and animal form urea in urine, found in urine or excreted in urine. Urine decomposes in soil, decomposes in nature. Decomposition of animal and the plant matter to form ammonium ions. The flesh or the corpus of animal and the remaining part of plant, even leaves, fallen leaves, what would happen? It decomposes into ammonium ions, NH4. Okay, NH4 plus. Okay, what happens to NH4 plus ammonium ions? Nitrification, oxidation happens, occurs to convert ammonium ions to nitrite, then nitrate. Nitrite firstly, then nitrate ions. Okay, the process of space occurs, returning nitrogen to the atmosphere. How this process? Take the nitrogen to the atmosphere, lock to the atmosphere again, called denitrification. Denitrification. Okay, this summarize the nitrogen cycle, the whole nitrogen cycle. I would like to make a revision of this nitrogen cycle by this chart. Look please. Firstly, nitrogen in the atmosphere. Here, nitrogen fixation takes place by bacteria, both soil bacteria and bacteria in plant roots, root nodules, to form ammonium. Okay, ammonium nitrification process, nitrification by bacteria, nitrifying bacteria. Okay, have form nitrite, then oxidize, extra oxidation to nitrates, NO2 minus 1, then NO3 minus 1. Okay, nitrate is absorbed by plants. Nitrates inside plants as source of nitrogen enables the plant to make proteins. Okay, eaten by animals. Both plants and animals died, or their waste in soil. They are organic compounds containing nitrogen, decomposes by decomposers, by fungi, by certain types of bacteria to produce ammonium. Okay, this is called ammonification, not important. Okay, to produce ammonia. The only route, the only way, pathway where nitrogen returns back to the air through certain type of bacteria found in mud okay, in soil locked with water, okay, in anaerobic conditions, this bacteria return back nitrogen into the atmosphere through the process called denitrification. Denitrification. It's okay? Okay. State the names of two processes that occur in both the carbon and the nitrogen cycle. Two processes share in both carbon and the nitrogen cycle. Let's see carbon and the nitrogen cycle. Look, CO2, okay, this carbon cycle, this nitrogen cycle. What's the similarity, what's the sharing points between them? Here, look, when animal feed upon plant, like cow or herbivore feed on grasses or trees, tree leaves. Look also here, this feeding, this sharing process. Look, this feeding, sharing process. What else? The composition also. Look at the composition. Animal plants, after death, all their remains, their waste, decompose by bacteria or fungus to form ammonia here and here also. Okay? Okay. Now state the principal source of energy input to biological systems. The main or principal source of energy is the sun. Look, this food chain driven or powered by the sun energy every day due to photosynthesis process must be done to allow or to provide food for herbivore. Then the cycle move along the chain. It's okay? So the sun is the principal source of energy input to all biological systems. Yes, never give up. All my best wishes for all of you. Thank you. See you next exam, inshallah. Bye bye.